Thank you so much for the opportunity to speak. I think I couldn't have asked for a better question to go into my presentation, because my experience is sitting in the radiopharmacy in a hospital and having to say radiochemical purity and all of these things. What are we actually injecting into the patient? And I think sometimes you feel you're in between the clinical picture and you're in between the research community. So disclaimer, I'm a radiochemist / radiopharmacist. So in the past, I was a radiopharmacist in the clinic, but now I've moved more into research and I keep myself busy with chemistry. But for today, I will put my radiopharmacy hat on and just speak from that perspective. So I challenge the audience to pick my favorite isotope after this, and I hope you cannot. I hope I make everybody equally angry, I guess. So when we are in the radiopharmacy, we have to look at the alpha emitters available. We have to be able to quantify the concentration and amount that we are using, maybe the molecules or whatever way you want to say that. We have to say what we are injecting radiochemically for yield and purity, and then also look at stability, waste management, and then the health physics part. So literally standing on the interface of all of these important questions. And while it was maybe in the past a bit more clear-cut with beta minus emitters and all of the other isotopes, it is quite complicated with alpha emitters. And we are standing between people that want to quantify exactly, and in the clinicians that has the patient in mind and the risk benefit, because in the end of all of this, there is a patient with a clinical need. And we have to balance these two opinions out and find out what is the best way to go forward. So for my presentation, I took from this article, maybe the Hopeful 8. So I have this little theory in progress, and I'm still working on it. So next time you see me present somewhere or talk to me, you'll maybe see some different ideas about this. But I tried to also rate them on a scale of what is my favorite or not my favorite, or what are we working with. And I came up with this little chart because there is not a one size fit all. There is not one radionuclide that stands out. Everyone has its negative side. So again, we say this session is on problems. So I will be focusing on the problems. I do love them and I think we do need them, but we have to also be open-minded about their maybe the lacking areas. So I'm just going to run through this quickly. I don't have that much time to talk, but I start with astatine, and we can see that there is some issues with production. You can have issues with the chemistry, and if I say stability in this aspect, astatine is quite stable with its one alpha emission or maybe just a short decay chain, but there is also chemistry involved, and the chemistry for smaller molecules is not always that good characterized with astatine, and I will get to that. The operator safety, because we are dealing with a halogen. So, you have certain things that you have to keep in mind, but measurement, maybe it's a bit better than the others. Actinium, a very good chemist friend of mine actually induced me to say it's a 2 on the scale, but I think it's maybe a bit more. But yeah, we have DOTA chelators. It's working, and you can have a centralized pharmacy because of the half-life, so you can make actinium and you can ship it out perhaps if it's stable, and that's why I also gave it a lower scale on stability, because we don't know. The measurement, of course, you cannot directly measure actinium, and operator safety, it's always something to consider. Bismuth, very nice and everything, but you cannot make it centralized and ship it out. It has such a short half-life, and the production capacity is, of course, dependent on the actinium availability. Late 212, suddenly it's all over the place, and it sounds really great. And yeah, there is also things to consider there: chemistry, stability, operator safety, centralized radiopharmacy. And please don't hammer on these numbers later. I said it's a theory in progress, so I want you to communicate with me where you think I'm wrong. But yeah, it's just the idea. Radium chemistry, what can we do with radium in the chemistry sense? Not that much. Terbium, the production, yeah, I can go on. And thorium, I don't know that much. So if you're working on thorium, please talk to me a little bit as well. I want to hear your opinions about that too. So clinical trials, I think there is better figures out. I saw now an E&M article published on clinical trials and how we go forward with alpha emitters and actually all radiopharmaceuticals. But this was just me looking at clinicaltrial.gov and seeing where are we, which ones are the most used. And I was quite surprised to see that thorium is actually showing up. even though I know very little about it. Actinium is there, astatine, bismuth, one trial registered. So it's gaining momentum. And then I come into the alpha radiopharmacy. So I feel we are like these rabbits. Maybe the one person think it's a very little issue, but it's actually very underestimated and it's really actually a big problem. And then the other group says, oh, no, it's really a big problem, but maybe it's not that much that we cannot use it. So this is where I am in between. I don't know. But I feel you do need a superior infrastructure, superior training. Your staff needs to know what they are working with, and we are having unique detection systems necessary. So for us, when we do research in these high infrastructure environments or in the company, it's very easy to say, you know, this works very well in my setup. But I want to urge the audience to look at where are we ending with this, because you make your actinium and you make your lead generators and you say, okay, I can do all the QC I need in my setup, but you might ship it out to people in very low infrastructure environments and low income countries. And I've seen this happen. And then they don't have the necessary skills or the understanding to know what they're working with. So we have to also discuss this at some point. So first of all, the problem in the pharmacy with alpha emitters is that you measure low activities and you sometimes use indirect measurements. And the dose limits are said to be, if you misadminister, if you have a difference of the prescribed dose by 20%. Maybe it's different in different countries. But if you look at something like actinium-225, it can be from 4.8 to 7.2 megabecquerel difference. And this makes quite a big difference in the treatment of the patient in the end. So the dose measurements has to be extremely accurate, and we sometimes work with very low activity. So you have to calibrate your dose calibrators or your measuring equipment. And inside your facility itself, you have to validate also your equipment with one another. And I have this method here that was discussed with me by Eric de Blas from Erasmus MC, where they actually validate their gamma counter and their dose calibrator, and they have spreadsheets and they work out exactly how much to draw and give to the patient in the end. And they really cross-calibrate every single one with a validated actinium source as well as one another. And this is the kind of technical things that you have to take into consideration in your radiopharmacy. So then I'm coming to what we understood as GMP production. And I'm going to use a very nice example of lutetium, because I think lutetium has been done quite extensively and we understand how it works. And if you want to test for radiochemical purity, you will normally, and I wonder if I can get a little laser pointer there. You would normally do a cold reference standard, where you have a non-decaying isotope that has a similar chemical properties like a natural lutetium, and then you will label it and you will characterize it with techniques that can clearly exactly tell you the mass of this molecule, how it looks, the structure, everything. And then you will validate your analytical methods, in this case, HPLC, with this molecule to see exactly what is the characteristics of this compound. So you have a chemical structure that you then translate into a chromatography technique, and you know exactly where it comes out. You have your labeled compound, your unlabeled compound, and then you radio label, and then you link the characteristics of your cold construct to your radioactive construct. Now, for these isotopes down here, we don't have cold isotopes, so we cannot do this. For actinium, this is quite problematic because we assume some things about the QC of actinium because of the way the gallium compounds behave, or the lutetium compounds, because we don't have a stable actinium isotope available. This is also true for astatine. Then actinium is in more trouble. So we have a lack of useful gamma emission. So we have to measure the daughter isotope. So this becomes even more complicated with the radiochemical purity, where you would do an ITLC strip or any chromatography technique. You will develop it, let it stand for 45 minutes until the daughters has been given off in a certain equilibrium. And then you measure it in your equipment, and then you determine retrospectively what the radiochemical purity was 45 minutes ago or even longer. And there is some methods being developed for radio HPLC, but this becomes even more tricky because you have very limited amount of activity that you can safely do with these systems. So you have to also then validate your system in low activities, and you have to do fraction collection and measure it. So the QC of actinium radiopharmaceuticals is a very tricky system indeed. And if you measure at the wrong time point, you get the wrong purity or yield. For instance, we tend to now even say this is not radiochemical purity that we measure with the ITLC strips. This is just the yield that we measure because we cannot really declare the purity. So in essence, you will then wait 2 hours perhaps, and then you will measure this, and then you will know what is the yield or the labeling efficiency that you had. Now, previously the EANM guidelines stated we should also validate the limit of quantification, and you should be able to say with 0.5% what is the impurities in your mixture. So you can imagine that this is virtually impossible with these methods. And then, of course, HPLC method recovery is also important because you need to know if you have any system, is there any radioactivity that stays behind on your columns or the things that you use to test the purity with. And with these low amounts, this is something we really cannot say. So yeah, it's maybe a bit too much to go in for this audience, but there is really a lot to do with the QC, and if it's even possible to have a GMP quality control for most alpha emitters that is equal to that we are used to with lutetium and all of the other beta minus emitters is still the question. And I guess when we look at the clinic, we should also start asking, is it needed, or what is the benefits and risks? And we have to weigh up all stakeholders in this discussion. So radiopharmaceutical stability. So as we see, there is many half-lives, very long half-lives, very short ones to choose from. You've all heard about recoil energy. So the amount of energy given off by this alpha emission can maybe, if this is captured in some chemical form, it can actually release the alpha emitter from the vector that you use. This is especially important with the alpha emitters with these long decay chains, because not only are we changing the chemical entity, so it might be that it decays from an from a metal radioisotope to some halogen or something else. And then it will definitely not be chemically compatible with the chelator, but we are also talking about high energies and other factors. And then also just pure chemical stability, as again, I'm going to say, sorry, astatine, but this is a well-known problem for that. So yeah, we have a stability issue. It was proven with beta minus emitters that you get radiolysis and all of this. I will show this a little bit later. So you cannot tell me that this is not an issue with an alpha emitter, and we have to make sure that we can determine that these radiopharmaceuticals are stable. So the chemistry of the daughters change, like I said. So you start out with a certain metal isotope, but then later on in the decay chain, the chemistry is completely different. So indeed, if you do radiochemical purity, you cannot say that the radiopharmaceuticals later on is actually the actinium product that you have, for instance, or lead-212 or whatever is the matter. I hear that lead-212 is suspected to have a little bit less of a problem just because of the half-life that we are talking about. It's not 10 days. And then again, thorium-227, not much is known about that from my side at least. So I think this is also a problem there. The detection of radiolytical products is important. So I have here a very good publication by the Erasmus MC group, where they actually showed with lutetium PSMA that there is a certain amount of radiolytical products that wasn't picked up by normal radiochemical purity measurements, but that it in fact had quite a substantial effect on the uptake in the receptor where the radiopharmaceutical has to go to work. And we talk about very little amounts, like 4%, 3%, and then that has an effect. So we don't have the resolution capabilities with current radiochemical purity determinations of alpha emitters to pick up these small changes. Shipment and shelf life. This is a question for all the pharmaceutical companies. Your radionuclide has a certain shelf life, and if you have worked with alpha emitters and labeled with them before, you know that many of them have constraints about radio labeling yield going down the later you receive the product. They do have shelf lives. It's not necessarily linked to the half-life of the radionuclide. It can be the buildup of contaminants from the vials they are shipped in. It can be just some of the daughter isotopes. It can be many things. Also, how you prepare your radionuclide, if you're shipping it in solution or if you're shipping it in powder, has an influence. Then you label it. So there, of course, you purify and do whatever you need to do, but then your product also has a shelf life, and this is to do with radiolysis, recoil energy, all of those things, as well as just vector stability. It's endless. Also, the physical half-life and how long your radionuclide lasts. So to say actinium has a 10-day half-life, it's brilliant. I can ship it for 3 weeks or 4 weeks. That's really not the case. Many good institutions I know haven't been able to determine the shelf life of actinium radiopharmaceuticals longer than a day. So this picture I took from LinkedIn. I'm very avid on LinkedIn, so please connect with me. I like to talk and see what everybody's busy with. And they shipped, and I removed all the visible characteristics of where this is from. I'm not naming or shaming at all, but they have a shelf life here that was very interesting. As you can see, it was produced on the 12th, and its expiration date is on the 19th. Of whatever year that is, I'm not sure, 1402. But the issue here is these products are out there, they are shipped to people. And according to my knowledge, I've not seen any single scientific publication or report that shows stability of these products beyond 1 or 2 days. So I want to know how this is possible and how this is allowed. So if you have your radionuclide, you are producing it, you have to now choose, are you going to centralize your labeling and ship out the final products to everybody? Or are you going to ship your radionuclides to them? Or are we going to look at generator systems and also what is the shelf life? And all of these things are really important for the community to consider. Waste management. I'm getting to the final part of my talk. So I have here the different productions of actinium. I think we have discussed them before, but each of these methods contribute indeed some part of the actinium need, and I think we all need all of them. But when you use actinium, you have to be sure where it comes from and what is the impurities that you have and what is the influences on your waste management. And it's far beyond just waste management of your production. It's also what happens when it's injected into the patient and the waste of the patients. Do you need to have them keep the urine or whatever that is excreted out in bottles and give it back to the radiopharmacy? And it sounds like I'm making a big deal out of this, but lutetium-177, not too long ago, this was a big discussion. Because you could get lutetium that is carrier added, and it has the metastable lutetium contamination, and you got lutetium that is non-carrier added, and that doesn't have that problem. And this was a discussion, and if you think back on that, this was really something to consider, and I think for actinium, this is also really important. Okay, yeah, like I said, I was still publishing this, and this is 2023. So lutetium metastable is still in consideration. So waste management problems can be from long-lived impurities from production methods, and also parent radionuclides that come out in your generator systems. And one example that resonates with me from my hospital days was the germanium generator that had some germanium breakthrough, and how we had to store germanium waste for years. So yeah, this is really important for the pharmacists sitting in their pharmacy, wherever that might be. Health physics and safety. I'm sorry, late people. I have to ask this question too, because I am now recently, I became aware of lead-212 and it sounds really interesting and great. And we have a gold standard to measure and we can image and it's really exciting, but upscaling might be a problem in a hospital radiopharmacy setting because there is a spelling error, but maybe the exposure to the radon gas or the shielding of the generator, I want to know how this will happen. And whenever I ask this question, I'm being told, oh, you just put figure lead on. But if you are sitting in your hospital radiopharmacy in a country where this is maybe not so easily accessed, to say, just put in a hot cell or a lead shielding, it's not always feasible. And I wonder sometimes if this will actually be done and if we will end up with operators being exposed beyond what is really a LARA. So if you design these things, please also think about the end user and don't think about your most optimal end user. Think about maybe your least optimal, because are you going to say this infrastructure is necessary before I sell my product to you, or are you just going to sell it and say this is not my responsibility? Shame. Per astatine, I also have to mention again with the halogen, this is also dangerous. So yeah. Useful reads. So all doom and gloom is past now. I think I really like this article and I want to highlight it. It's about more GMP-like practices for actinium radiopharmaceuticals by the group from Erasmus MC, and I think you can definitely have a look at a more practical example of how actinium-225 will be labeled in a radiopharmacy. They have excellent ideas. This is me. I am a fan of actinium, but there is also the questions of the QC, and I went into great detail of the quality control. As I discussed very little here, you can read up if you want to know more about that. I think Richard Zimmerman gave us some quite good ideas about the upscaling of lead-212 and actinium, if it's also really happening. And it seems that we can end up with great capacity of production of these two isotopes, because indeed, if you don't have the isotope, why do we even care? And yeah, also Valerie and co-workers have a general state-of-the-art article about all the production opportunities, which is also a great read. Then I'm going to give an extended version of this presentation and also add some positive things about each. In my presentation at the European Symposium of Radiopharmacy and Radiopharmaceuticals in Coimbra, but also I know everybody cannot maybe attend, so I will make sure that there is a recording afterwards available that you can maybe ask if you want or if you're interested. And I have too many people to thank, so my ideas here is not practical examples from my own practice, but it's based on scientific conversations I had with many people over many times, and it's my knowledge is growing, and I try to add more when I hear. So if you want to enter a discussion with me at some point, please link up with me on LinkedIn, and yeah, I would be very interested in your opinions. Thank you.